Never stop trying, never stop believing, never give up. Your day will come. Welcome back to our channel. Today's lecture is on a very important topic, that is gram stain. This question is asked in your theory exam as well as in your practical exam. So watch this lecture carefully. Now to begin with, the study of bacteria can be done by two ways: by preparing unstained preparations or by stained preparations. No, now these unstained preparations are examined mainly for the bacterial motility and for the spirochetes. While the stained preparations, they provide color contrast to the bacteria and so they become visible in the microscope. The smears which are made by bacterial culture are first dried and then they are fixed by heat. This heat, it coagulates the proteins in the bacterial cell and fixes the bacteria on the glass slide. Now, this fixed slide is then stained with appropriate stains. What are the various common staining techniques? First, simple staining. Simple staining include use of basic dyes such as methylene blue or basic fustian. Now these simple stains have a disadvantage because they give all the bacteria the same color. So here in this picture you can see simple staining with methylene blue in which all the bacteria are stained blue. Now second type of staining is the negative staining in which the bacteria are mixed with dyes such as India ink or nigrosin, as a result of which the background, it takes the color of the stain while the bacteria remain unstained. This is very useful for the demonstration of bacterial capsules. So here you can see the negative staining in which the background appears dark and the bacteria is stainless or colorless. The third type of stain is the impregnation method of staining. It is used for the thin bacterial cells which cannot be visualized under microscope. So these thin bacterial cells are thickened by using silver. The bacterial cells are impregnated with silver. This makes the bacterial cell visible. Now, the bacterial cells are mostly the flagella and the spirochetes can be visualized by impregnation method. So here you can see silver staining used to visualize these bacterial structures. Now the fourth type of stain is the differential stain. As the name implies, the differential stains impart different colors to different bacteria. Examples of differential stains include gram stain, acid-fast stain and the Albert stain. So here you can see the differential staining of two types of microorganism. One is stained blue while the other is stained red. So a simple picture to remember this. The simple stain has given all the bacteria same color. Negative staining in which the bacteria remain unstained while the background is stained, and differential staining, as the name implies, different colors are given to the different bacteria. Now let us study the gram stain in detail, which is a very important topic asked in your viva as well as theory. So gram stain is the most widely used stain in bacteriology. It was originally developed by Christian Gram in the year 1884 and it is a method of staining the bacteria in tissues. Nowadays, certain modifications of gram stain are used. So, to remember the entire process of gram stain, let us divide it into 7 easy steps. So, in step 1, the smear of the bacteria is heat fixed. So, heat is applied to this smear and it is fixed. Because of the application of the heat, the proteins in the bacteria are coagulated and the bacteria is fixed to the glass slide. Now the step 2 is that of primary staining. The primary staining is mostly done with crystal violet, which is also called as the primary stain. And This stain crystal violet is applied for 1 minute. The other alternatives to these crystal violet are gentian violet and methylene violet. Step 3 is to pour grams iodine on the slide. This grams iodine is nothing but dilute solution of iodine, and this is also applied for 1 minute. Step 3 is to wash the slide with water. Step 4 is to wash the slide with water. After primary staining and application of grams iodine, then the slide is washed with water. Step 5 is decolorization with the help of acetone, which is done for 10 to 30 seconds. Apart from acetone, you can also use alcohol. Then the next step, that is step 6, is to wash the slide again with water. And step 7 is counter staining with safranine. So safranine is used for counter staining, which is done again for 30 seconds. Other than safranine, carbol or neutral red can also be used. Now let's summarize this method. First step is heat fixing. Second step is primary staining with crystal violet. Then for 1 minute. Then comes use of grams iodine. The third step again for 1 minute. Then wash with water. That is the fourth step. Then comes the fifth step, that is washing with acetone for about 30 seconds. Then again washing with water. And the last step is counter staining with safranine again for 30 seconds. So this is the summary of gram staining. Now the gram stain, it differentiates the organisms into gram positive and gram negative. Now the gram positive organisms, why do they appear gram positive? Because they resisted the decolorization and they retain the primary stain, that is crystal violet. So the gram positive organisms appear violet under microscope. While the gram negative organisms, they get decolorized with acetone and they take up the counter stain, that is safranine. So they appear red in color. So here you can see the gram positive organisms that they have retained the primary stain, that is crystal violet, while here you can see the gram negative organisms that have taken up the counter stain, that is safranine. Now, what is the mechanism of this gram staining? Gram staining is attributed to two factors, the permeability of the cell membrane and the integrity of the cell membrane. Now, talking about the permeability of gram positive organisms is different than gram negative organisms. How? Let us see. Now, the gram positive organisms have a more acidic protoplasm. So if this is a gram positive organism, the protoplasm of this organism is more acidic. So the primary dye, crystal violet, which is a basic dye, is strongly taken up by this acidic protoplasm. Secondly, the mordant, that is iodine, which is used, it makes this protoplasm more acidic, and a complex is formed between the primary dye and the iodine. This complex is called as the di-iodine complex. So when first the bacteria, it takes up the primary dye, then it takes up the iodine. So a complex between the dye and the iodine is formed. Thirdly, the gram-positive organisms have a less permeable cell membrane. So this di-iodine complex is entrapped inside this cell. So they resist decolorization because this membrane is less permeable. Now, the gram-negative organisms have an increased permeability. So, when the decolorization is done with the alcohol or acetone, there is rapid intake of alcohol inside the cell. And because of rapid intake of alcohol inside the cell, there is also rapid outflow of the diiodine complex out of the cell. So the gram-negative organism, they loses the diiodine complex on washing with acetone or alcohol. Second attributable factor for gram staining is the integrity of the cell membrane. The integrity of the cell membrane is essential for gram-positive staining. If damage occurs to the cell membrane, even the gram-positive organism loses the diiodine complex and appear gram-negative. So here you can see in a simple picture the gram staining process. Suppose this is a on this side you can see gram positive staining, in here you can see gram negative staining. Both the specimen are fixed, both take up the crystal violet color. Upon iodine treatment, the diiodine complex is formed. Upon decolorization, gram positive organisms, they resist decolorization, while gram negative, they undergo decolorization. Upon staining with safranin or counter staining, the counter stain is not taken by gram positive organisms, while the gram negative organism, they take up the safranin or the counter stain and appears red or pink. So this is the basic science behind the gram staining. The next lecture will be on Ziehl-Neelsen stain. So a question for you today is, what kind of stain is gram stain? Do let me know in the comment box. Is it a simple stain, or is it a differential stain, or is it a negative stain, or it is an impregnation stain? Let me know how much you have understood from this lecture. Leave your answer in the comment box below. That's all for now. Happy studying.